Welcome to AG Biology. Today in this video we discuss about mitosis under the chapter Cell Cycle and Cell Division for Class 11 Biology. Firstly, you might have already heard about cell theory. What does it say? It says all living organisms are composed of cells and these cells arise from pre-existing cells. So, cell division becomes very important for all the living organisms. What constitutes a cell cycle? Or what's happening in a cell cycle? The cell prepares for its division by means of DNA replication, synthesis of other components, and finally divides into two daughter cells. So it is a sequence of events by which a cell duplicates its genome, that is DNA, and synthesizes the other components of the cell and eventually dividing into two daughter cells. So now we will see this mitosis which is happening in the somatic diploid cells in detail. What is the other type of division that you might have heard of? Which is happening in germ cells responsible for production of gametes? Yes, that is meiosis. Mitosis is happening in somatic cells whereas meiosis is happening in germ cells. And during mitosis, you can see the number of chromosomes of the daughter cells and parental cell remains the same. That is why it is known as equational division. On the other hand, if you look at meiosis, the number of chromosomes in the daughter cell is reduced into half. So there is a reduction in the chromosome number. That is why it is known as reduction division. Okay, so mitosis is equational division, whereas meiosis is reduction division. So we will come back to the mitotic division. So take a typical human cell. The duration of the cell cycle is 24 hours. Does it mean all cells in the body dividing at the same rate? No. Actually, there are slowly dividing cells like heart cells, yes, liver cells. On the extreme hand, you have nerve cells which take years to divide. Also, if you look at simpler organism, the division time is much lesser. For example, a single cell eukaryotic organism like Saccharomyces cerevisiae, which is a budding yeast, which takes 90 minutes to divide. So, the duration of cell cycle depends on two factors. Within the organism, the cell type, it varies. Also, between organisms, the duration of cell cycle varies. But as an average human cell, you can take the duration of cell cycle as 24 hours. So the cell division of an average human cell is divided into two. One is interphase and second one is M phase. What is interphase? Interphase is a period or the phase during which cell prepares for its division. How cell prepares for its division? It duplicates its DNA, it synthesizes proteins, and cell growth happens. And what is M phase? M phase is a phase during actual division is happening. Interphase can also be explained as the time period between two successive M phases. So if you look at M phase, this can further be classified into two. One is karyokinesis and the second one is cytokinesis. What does karyon means? Karyon means nucleus. So karyokinesis is nuclear division, whereas cytokinesis is the division of cytoplasm. So for convenience, this karyokinesis can further be divided into different stages, namely prophase, metaphase, anaphase, and telophase, that we will see. Now let's look at the interphase. What is interphase? It is the phase during the cell prepares for its division by means of DNA replication, components synthesis, and protein synthesis. Yes. This can be clearly demarcated as G1 phase, which is gap 1 phase, S phase, that is synthesis phase, and the last phase is G2 phase or gap 2 phase. During G1 and G2, the cell growth happens, or you can say cell growth is kind of continuous during the interphase, entire interphase. But synthesis phase is the only phase where DNA replication happens. So DNA replication is limited to only one particular phase of interphase, that is known as S phase. Another event that happens in S phase is centrosome duplication and protein synthesis. This is a synthesis phase. Now we have to be clearly distinguish between haploidy number and DNA content. For a typical eukaryotic diploid cell, at the start of the G1 phase, the haploidy, which is the number of chromosome, can be denoted as 2n. And the content of the DNA can be denoted as 2c. What happens during S phase? During S phase, the DNA content is getting doubled, or the sister chromatids are formed. So the haploidy number remains the same, 2n, but the DNA content remains 4c. So throughout G2 phase, which is just after S phase, what will be the haploidy number? Since it's equational division, there is no change in haploidy number, which is 2n and 4c. That is the DNA content. We will see some questions based on this DNA content and haploidy while discussing questions. So then, what is quiescent state? We already said not all cells dividing at the same rate or 24 hours. There are some cells like heart cells, they divide very rarely. So when there is a need of repair, these cells divide. So these cells are in kind of an inactive state. From G1 phase, they exit and comes into a inactive phase of gap phase, which is known as G0 phase or quiescent state. So here the cell is metabolically active, but it is not proliferating. So M phase can be divided into two. One is nuclear division, that is karyokinesis, and then cytoplasm division, which is known as cytokinesis. So the karyokinesis is kind of continuous, but for convenience, you can classify this into stages. The first phase is known as prophase. Pro means first. Remember that way. What happens during a prophase? The chromatin condenses into chromosome and sister chromatids started visible. Okay. And then centrosomes, these are synthesized during which phase of the interphase? S phase. And these centrosomes move into opposite poles. And these radiate out, and along with this spindle fibers, you call them as mitotic apparatus. We will see this. And during the end of prophase, all these membrane-bound organelles like ER, Golgi, nucleolus start disappearing. And nuclear membrane is also invisible during the late prophase. Second phase of karyokinesis is known as metaphase. What happens during a metaphase? The chromosome condensation is completed. That means you can clearly see sister chromatids, one here, second here, attached together at the centromeric region. Okay. The centromeric region, you can see another disc-shaped proteinaceous structure, something known as kinetocosis, to which the spindle fibers are attached. So spindle fibers are attached where? At the kinetocosis. So kinetocosis attaches one chromatid to one pole of the spindle fiber and the sister chromatid to the opposite pole. So as the time progresses, you can see these chromosomes lie at the equator region, and this arrangement is known as metaphase plate. The metaphase is marked by metaphase plate arrangement. Meta meaning between. Remember that way. Next phase is anaphase. During anaphase, what happens? The centromeres get split. So the two daughter chromatids are separated right now and they move towards opposite poles. Since the centromeres are split, how will you count the number of chromosomes? Just count the number of centromeres. So here the number of chromosomes has been increased. Let's look at the ploidy and DNA content during different phases of karyokinesis. Yes, as genome has duplicated during S phase, the DNA content can be described as 4C and the ploidy level is 2N. It is the same during metaphase, 2N and 4C. What happens during anaphase? The daughter chromatids are separated and you can see the ploidy number is 4N and the DNA content is 4C, which is the same for telophase. What happens during telophase? Telophase is the end of the karyokinesis. Telo meaning end. What is the sequence at the end of the chromosomes? Yes, that is telomere. Telomere is the sequence found at the end of the chromosome. Telophase is the last phase of karyokinesis. So in telophase, what happens? It decondenses to chromatin, right? So all these ER, Golgi, nucleolus, nuclear envelopes start disappearing at the late prophase. This will start reform at telophase. Okay, the ploidy level and the DNA content will be the same like 4N, 4C. Is the division completed right now? No. It's only karyokinesis have been completed. So there is actual division of cytoplasm which is known as cytokinesis. There is a difference in the way which it is achieved in animal cells versus plant cells. Okay. In animal cell, a furrow happens at the membrane and it deepens and finally joins at the center and division of the cytoplasm happens. But in plant cell, it happens in the opposite direction. Plant cell, remember, they have a tough cell wall. So the cytoplasmic division happens at the center and it joins towards the lateral cell walls. Something known as cell plate. And finally a new cell wall is formed. What will be the ploidy and DNA content? So it will be 2N, 2C here, 2N, 2C here. As we see, mitotic division is also known as equational division. There is no change in number of chromosome number, which remains at 2N. Now we will examine some of the questions that being asked in the previous year. In S phase of the cell cycle, the amount of DNA doubles in each cell. The amount of DNA remains the same. Chromosome number is increased. Amount of DNA is reduced to half. What is S phase? S phase is a synthesis phase. So during S phase, DNA replication or duplication happens. So the DNA content remains double. It gets double. But the ploidy remains the same. Ploidy remains the same. So number of chromosome is increased, wrong. Amount of DNA remains the same, wrong. Amount of DNA get doubles in each cell. Answer is A. During mitosis, the ER and nucleolus begins to disappear. At which stage? We have already seen it is during late prophase. Late prophase. Okay. Which of the following is not characteristic feature during mitosis in somatic cells? Spindle fibers. Of course there is spindle fibers. It attaches where? To the kinetochores. Remember that. Disappearance of nucleolus starts at which phase? Prophase. Late prophase. Precisely late prophase. Chromosome movement. Backward movement happens at which stage? Anaphase. Remember, ana means backward. So anaphase is characterized by backward movement of chromatids towards the opposite poles, right? Synapses. We haven't discussed this so far, which is happening in meiotic cells, in meiosis. So, which is not characteristic feature of mitosis in somatic cells is synapses. Spindle fibers attach onto telomere of chromosomes. What is telomere? It is the ends of chromosome. No, spindle fibers doesn't attach there. Centromere of chromosome? No, not to the centromere. The centromere, you have a disc-shaped proteinaceous structure known as kinetochore. It is not kinetosome, it is kinetochore. So, spindle fibers attach onto kinetochore of the chromosome. During which phases of cell cycle the amount of DNA in a cell remains at 4C level, the initial amount is 2C. Initial amount is 2C, that means at G1 it is 2C. Okay. What happens at S phase? DNA duplication happens, so it will become 4C, but after S, right? After S it will become 4C. Throughout G2, it remains at 4C. The DNA content will be 4C. And during M phase, yes, during different phases, there is change in ploidy and DNA content. For example, cytokinesis. Cytokinesis, it is 2N2C, right? So what is the answer? Yes, only during G2 phase it remains at 4C. So only G2. G0 and G1, no, it is at 2C. G1 and S, G1 it is 2C. Yes, it is start getting 4C, but after S it is 4C. Only G2. G2 it is 4C. G2 is right, but M phase, throughout the M phase it is not 4C. For example, after cytokinesis it becomes 2C.